गुड इवनिंग नाबिया राज। गुड इवनिंग। आज के लाइव में अगर आप अटैच हो चुके हैं तो काइंडली गिव मी मैसेज बिकॉज इट इज नेसेसरी टू कंफर्म दैट सम स्टूडेंट्स आर समन व्हाट नाबिया राज आज ऑनलाइन का एसएमएस नहीं आया है। टुमारो yesterday I posted that today's class today. I just continue the class related with the genetic polymorphism, and in this live class I confined myself to the significance of genetic polymorphism for the evolution. Of course, I will take one class further on the topic of the genetic polymorphism to represent its different aspect. But it is my second class on the same topic, that is the genetic polymorphism. But today my class is associated with the role of genetic polymorphism in evolutionary the role of genetic polymorphism in evolution. In last class, we all discussed that the different form of gene present at any particular locus in gene pool is represented as genetic polymorphism. So it is simply we can say that the various phenotypes and variations in different organisms are evolved due to the genetic polymorphism. And actually, we studied the different components of the gene, and as I suppose, the next 30 to 40 years, or more than the 30 to 40 years, we will work on the different aspects of the gene, or the topic related with the genomics. So, the genomic is now a growing field. So, I just concise the informations related with the role of genetic polymorphism in evolution. We all know very well that the gene is actually a part of DNA, and the DNA was chemically explained in 1953 by the Watson and Crick. And the Watson and Crick explained that the different components, structural components, and the functional significance of the chemical organization present in the DNA, particularly the 2-deoxyribose, nitrogenous base, and phosphates. And now the it is the part of nucleic acid. The other component is the nucleic acid is the RNA, and after the reporting of the DNA or the after the reporting of the chemical nature, organizational pattern, and structural significance of the DNA by the Watson and Crick, a tremendous work has been completed in genetics and genomics. Up to date, a tremendous work is have been completed that lead to understand the different functional and structural or evolutionary significances of the DNA. But at this stage, we know many things related with the DNA. But it is also true that we don't know much more about the DNA, and some more works for the under proper understanding of the DNA and genes or genomics are required. So now it is a hot topic, and the globally it is a hot cake for the research and for the students of the PhD. Of course, some common persons are also interested to know about the genomics, because in all the physiological activities, inheritance pattern, disease, and other physiological and metabolic activities, all are controlled by the gene. So it is a burning topic of the research, and it will continue up to the more than the 30 to 40 years further. Chemically, we all know that chemically the DNA is formed by the sugar, phosphoric acid, and nitrogenous bases. And popularly, the DNA is referred as the molecule of life. It is referred as the molecule of life. It is a type of nucleic acid present in the cell nucleus, particularly in the eukaryotic cell, and function as a storing and transferring the biological information throughout the generations. That is, it is inherited genotype. Now, the number of works, because today I confined, not confined, but I emphasis on the human, the role of genetic polymorphism in evolution, with emphasis on the human beings. So, throughout world, the Human Genome Project released that the genome, what is genome? Genome, a compilation of all the DNA of a cell, is made up of some functional DNA sequences. The complete DNA or haploid, if you use the terminology, then haploid set of the chromosome of any cell represents the genome, and the DNA is present in the chromosome. So a compilation of the all DNA present in a cell is made up of some functional DNA sequences and useless DNA sequences. Of course, all DNA present in any nucleus are not functional. Some are functional and some are useless sequences, because the DNA sequences are responsible for the functional aspect. Of course, the functional aspect is observed commonly in the term of nucleotide sequences, commonly known as the gene. But the components, all the nitrogenous bases sequences, or all the chain or stage of the polynucleotide chain present in any chromosome or in the form of DNA is not functional. Only a small number or a small quantity of the DNA is functional. Whereas the other components are non-functional. So the DNA which are functional, and they are responsible for the synthesis of protein, and that proteins have the expressive that proteins are responsible for the expression, whereas the some other DNA are useless and commonly referred as junk DNA. So, and if we estimate the percentage of functional and junk DNA, then more than 90% of the genome is present in the form of junk DNA, whereas only 10% of DNA of any genome is functional and responsible for the synthesis of the protein. Although some recent research findings suggest that the junk DNA performs a function to regulate the gene expression. Of course, the part of the junk DNA is not responsible directly for the synthesis of the protein, but the part of the junk DNA or some part of the some sequences of the junk DNA is responsible for the regulation of gene expression. So it is not true that we can say that all junk DNA have no molecular activity. Of course, some sequences of the junk DNA is responsible for the molecular activities, and that molecular activities are responsible for the regulation of expression of the DNA. So, the regulatory part of the DNA are known as expressive sequence of the polynucleotide chains. That is, another fundamentals proposed so recently that some components act as the epigenetics. We discussed the concept of epigenetics in present live classes. The chemical composition of DNA is same because it is formed by the nitrogenous bases, phosphates, and ribose sugar. Again, the nitrogenous bases are of two types, purine and pyrimidines. These components we all know. Uh, so the the chemical composition of the DNA are chemical composition of the DNA are same, but they differ in sequences of nucleotides or the particular nucleotidic sequence we observed by the method of the sequencing, and that particular sequence is represent variation from organisms to organisms. It is popularly from primitive. It know that they are same, and they carried from the primitive prokaryotic cells to the eukaryotic cells. And during the sequential movement from the prokaryote to eukaryote and from the primitive organisms to the higher organisms, the sequences represent variation. And that change is in our primitive ancestor guided the process of evolution. So, during the sequential evolution, the nature of sequences, of course, the chemically they are similar, but the nature of sequences, that means the arrangement of sequences, are responsible for the change as per the evolutionary event and during the evolution. Or in alternate way, we can say that the changes in the nucleotidic sequence is responsible for the sequential evolution from lower to higher animals. Of course, it is true, but the major questions are how, which factors are responsible for, how they are changes, when they are changes, what is the factors responsible for these changes, and all these how, what, where, when are associated with the evolutionary events. So that changes how, when, where, all the components responsible for the changes, and that changes and that answer of that type of questions provide information or knowledge for the development of the genetic polymorphism, and that genetic polymorphism can responsible for the evolution, and that genetic polymorphism is related with the evolutionary events. So, first, it is important to know that evolution of new species is not an event of one day, but it takes thousands of years, and during the thousands of years and thousands of years, the time span of the thousands of years is responsible for the formation of the species and formation of the higher group, and during all such long time, long period of time, the nucleotidic sequences or components are modified, and all that events a number of events are associated and that are responsible for the development of polymorphic nature of gene at any particular locus. Now, it is in last class I also explained that if you considered that the genetic polymorphism is defined as the change or alternation in the DNA sequences due to intrinsic and extrinsic factors that lead to change in the phenotype. And in my last class, I explained a number of events that are responsible for the development of the genetic polymorphism. And I am just increasing some more information in today's class. And it is my request that the students or the viewers who were not participated in my last class must go to my previous online video and again come and observe the facts and hear the informations provided today. So the number of events are associated for the development of the genetic polymorphism, and that genetic polymorphism is responsible for the evolutionary changes. They may be, the factors may be due to intrinsic or extrinsic factors. Intrinsic means the event or factor occurred within the individual or within the population. And the extrinsic factor, that means the factor associated with the outside, but means the surrounding. That means the both intrinsic and extrinsic factors are associated with the development of genetic polymorphism, and that genetic polymorphisms are responsible for the evolutionary events. And in last class, I represented a number of factors like the mutation, genetic drift, gene flow, gene migration, and among these, I represented in my last class that mutation, if any changes is observed in the arrangement and nature of the DNA sequences or nucleotide sequences of the any DNA, it is simply referred as the mutation. So it is also known as the mutation, that is the changes of the nucleotide sequences. The other factors which I related, the other events which are associated with the development of the genetic polymorphisms, as described in my last class, is the genetic drift, gene flow, gene migration, splitting of the population, selective marriage, objection of the randomized population, all these things. But the one important factor which I am discussing today and explaining today, that is the intrinsic, and one of the more important event responsible for the genetic polymorphism is error in DNA replication, which we discuss today. Of course, during the replication, it is responsible for the replication is responsible for the synthesis of the DNA, daughter DNA, and that DNA, synthesized DNA from the parent, is passes from one generation to the another generation through the gamete. But when the any error is observed in the replication, that may causes the genetic polymorphism, and it is most important intrinsic factor which is responsible for the development of the genetic polymorphism. And if we consider that error in the genetic polymorphism, error in the replication to develop the genetic polymorphism, we consider that if we study the molecular biology, then the repairing system is also present in the cells. And if we observe any error in any replicative daughter cells, that is the replication is a process of copying DNA. Simply put, in the daughter cells, in the replication, the enzyme which is responsible for the DNA replication is the DNA polymerase. And the DNA polymerase is synthesized by the DNA itself, because the DNA polymerase enzyme is also protein, and so it is synthesized by the DNA itself, and the sequence which is responsible for the synthesis of DNA polymerase is present in the DNA. And if the DNA polymerase does not work properly, so it is responsible for the development of error, replication error. And at the end of the replication, the process of polymerization slows down, which adds mismatch to the growing strand. The process is known as end replication protein. And when the end replication protein is either we all studied that SOS system is present, which is responsible for the repair of mismatch immediately. But still, it is unfortunate that sometimes some DNA cannot repair correctly. And in this way, the synthesized replicating sequence is responsible for the development of polymorphic sequence. And if the polymorphic sequence or the DNA synthesized with error is inherited to the next cell, it again comes in the replication cycle and considered as the correct parental sequence. And now it moves from one to another sequences, one to another cells, and from maternal to paternal sequence. Now, these sequences or the genetic polymorphism developed by the error-sum replication is analyzed by the other evolutionary factors or evolutionary forces. And if it will provide any advantage to the survival, in the survival rate of survivalship or in the reproductive fitness, it is selected, and now it moves from one to another generation. Now, a study during the last 2 decades, if we observe the studies occurred between the last 2 decades, explain that the environmental factors have a great impact. on the gene expression. For instance, if you observe in the population, human population, and consider the melanin or the expressive degree of expression of the melanin in population, the peoples living in the higher altitude and extreme temperature, while represented more expression than the peoples living in the colder region. Now the gene is same, but the degree of expression of the melanin synthesizing gene is more in the hotter climate than the cooler climate. The reason for this is the higher melanin coating is protects the skin from the harmful effect of the UV rays, ultraviolet, come from the sun. So it provides a survivalship benefit. And if it is protected, the UV light or the damage of the skin from the UV light, it is necessary for the increase of the rate of survivalship. Here, meaning thereby that the expression degree of expression is also associated with the polymorphic nature of the gene or expressiveness of the gene. And here, alternation of the gene expression, here it is the gene is same, but the degree of expressiveness is determined by the environmental factors, and that changes is referred as the epigenetic alteration. So I am saying just before that we discussed today is the epigenetic concepts also in the genomics, and then the epigenetic alteration, because the UV exposure in the warmer region is more than the cooler region, and the degree of expression of gene is related with the environmental factor. So here in the epigenetic alteration, the sequence of DNA cannot change, but still the phenotype varies. And now a number of research related with the epigenetic expressions or epigenetic alterations is associated with the study of the cancer causing gene, because there are a number of epigenetic factors have effect on the gene related with the development of cancer. So the phenotype gene is same, nucleotide sequence is same, but the expressiveness represent a variation in the with respect to the environmental condition, and it is referred as the epigenetic alteration. On the other side, if you consider the in different way, the polymorphism in gene sequences may be caused by the mutational changes or by the changes associated with the defective replication mechanism or different defective cell repair, gene repair mechanisms, because the DNA polymerase has the editing capacity. Itself is responsible for the editing of the wrong nucleotide pair if it is defective, or it is not is not in a position to edit the wrong pair if incorporated in the replication, then SOS mechanism is also responsible for the repairing system. In any way, if it exists, then it is the intrinsic, one of the most important factor which is responsible for the development of the gene polymorphism by the intrinsic factor, and that the changes is responsible for the development of the genetic polymorphism. For example, the single base mutation is the HBB gene, hemoglobin B gene, includes sickle cell trait. If we studied the sickle cell anemia, then at the sixth position, glutamic acid is replaced by the valine only by the change of the one nucleotide, and it is referred as the single nucleotide polymorphism, as we also discussed in my last class. And that mutation is observed as the beta chain of the hemoglobin at sixth position. Now, the sickle cell traits, of course, the sickle cell is the harmful to the population, and in the homozygous condition, it is responsible for the acute hemolysis, and in the heterozygous condition, it is responsible for the development of the hemolytic disorders in the condition of the oxidative stress. So it is lethal in the homozygous condition, but still it is present in the number of places. Why? Because it provides or it increases the rate of survivalship from the malaria. So in one condition, in one way, it is responsible for the it has lethal effect on the population, but in the another way, it is responsible for the it provides the survivalship benefit for the falciparum, Plasmodium falciparum. So, due to its rate, it is responsible for the increase of the rate of survivalship in a specific condition or in any specific geographical area where the rate of malaria incidence is more. So, the prevalence gene of the sickle cell anemia or the polymorphic nature of the beta chain is more prevalent in that particular area as in the Africa and in some conditions of the Southeast Asia. So the gene polymorphism is also present or represent the more prevalence if it provides the benefit of the benefit for the rate of survival. Now, when we condition, then how new variations or genetic polymorphisms occur. And we discussed a number of the topics in my last class, so it is not necessary to come again on the same topic for the development of the different factors. But if the genetic polymorphism occurs in the somatic cells, not in the germ cell, it remains restricted to some of the cells area or tissue of the body and is temporary. But if it observes in the germ cell, then it is inherited because the germ cell is responsible for the carrying of the genome from parental stock to the daughter stock. Then, contrary, if the genetic polymorphism occurs in the germ cell means in a sperm and egg, they are inherited. That is how the change or genetic polymorphism passes from offspring to organism, and it is help to the genome to express in consecutive generations. In this way, when the germ cell is affected by the component, then it is carried from one to another generation. The one again intrinsic factor, because in last class we have already discussed the extrinsic factors. So in today we discuss on the intrinsic factor. The another intrinsic factor is the transposon element, and transposon element is also referred as jumping gene or interspersed sequences. The concept of the jumping gene was at first represented by Barbara McClintock, and for that reporting in the May she got Nobel Prize. Now, the genetic polymorphism, if the genome is stable, but it is some segment moves from intragenic positions, translocate, moves from one to another segment of the chromosome, then the sequence is referred as the interspersed or transposon sequences, IS or transposon sequences. That particular transposon sequences is responsible for the formation of instability or creation of instability of the genetic material. For example, allo sequences present in the intronic sequences in the human chromosome. I am just talking for the human chromosome. And here the allo sequences move from one to another place. And now the movement of the transposons element are the mobile or moving genetic sequences. And that movement is responsible for the structural instability of the both intronic and exonic sequences. And if the transposon moves in the intronic sequences and affected the rate of splicing, then it is responsible for the changing of the nature of the mature mRNA, and in this way it is responsible for the changing of the nature of protein. In this condition, it changes gene expressiveness. In some time, we have information in number of places, particularly in the neurodegenerative cases also, or in the cases of the cancer, that some the interspersed or transposons moved in the exonic sequences. And when the interspersed sequences moved in the exonic sequences, it is responsible for the disease. And a number of diseases, some are also associated with the abnormal growth and the development of the cancerous growth, cancer cell, that is caused by the interspersed and movement of the transposons. So, the transposons may jump into regulatory elements or may jump into the exonic sequence. In any conditions, if they move in the intronic sequence or in the regulatory sequence or in the exonic sequence or in the intronic sequence, in any form, they provide intrinsic condition for the development of the polymorphic genes and responsible for the development of the genetic polymorphism. So here, the genetic polymorphism may be useful and may be harmful. And now The if the polymorphic genome or gene sequence moves from one to another population, it has the evolutionary sequences. And now the these components are associated with the movement of evolutionary cells. Now, the evolutionary concept, the genetic polymorphism, is also considered for the development of phylogenetic tree. Phylogenetic tree, that means the tree which represents the evolutionary steps, which represents the relationship among the populations on the basis of the evolution. Of course, I will again discuss on, I will give you one class complete for the proper understanding of the phylogenetic tree, because it is one of the significant part of the bioinformatics nowadays. And here I am just saying that the genetic polymorphism provides a basis for the development of the phylogenetic tree on the basis of the similarity or dissimilarity of the nitrogenous basis of within the same polymorphs, gene polymorphs. And the process of evolution and the changes or alternation in the DNA can be mapped by the constructing the phylogenetic tree. If what is the phylogenetic tree? The phylogenetic tree is a diagrammatic representation showing how different organisms are related. The phylogenetic tree is the best thing to describe or show how two species or organisms are different or related on the basis of the molecular similarities and dissimilarities. The construction based on the data of the genetic polymorphism of an organism, and means it represents that the closely situated species or organisms on the tree are more related and have less genetic polymorphism, and vice versa. That the two groups, if represented, the less similarity in the gene polymorphism, that means they are distant than the organisms which represent more similarity in the polymorph genes. So in this way, if we observe the gene polymorphism and we analyze the molecular similarities and dissimilarity of the gene polymorphism of the different organisms, we can easily develop or drive the phylogenetic tree for such related group of organisms. Now, the phylogenetic tree indicates that the relation between the organisms is inversely proportional to the genetic polymorphism and directly proportional to the less genetic polymorphism. So, here a common marker when we study the genetic polymorph, when we study the development of the phylogenetic tree on the basis of the polymorphic gene polymorph, then we consider the marker and the sequence structure selected and amplified by the PCR, then we study the similarities and dissimilarities. So, here we consider the next form. Now, the genetic polymorphism is evolutionary, is also associated with the rate of survival as I am saying just some time before that the survival is the primary goal of organisms, and if any polymorphic gene is developed or gene polymorphism is developed, has no role in the rate of survivalship with respect to the surrounding and environmental conditions, then the genetic polymorphism is not selected or not present in that particular population for a longer time, because that polymorphic gene is not carried from the one form to another. Let us take an example. What cancer is? Someone is consuming harmful things that are not at all right for the human body, like tobacco, and it causes cancer. Nature isolates the person by creating some sudden unusual genetic polymorphism, and literally tells us that isolate him, he is harmful to the entire population. And in this way, the changes in the gene, nature of the gene, is not selected by the nature because it is harmful. And another way, if we consider, just I am considering that the sickle cell anemia. Of course, the sickle cell anemia is harmful, but it provides the survivorship benefit for the disease, deadly disease, falciparum malaria. Now, the gene is selected. So here, if the polymorphic nature of the gene is developed in any population, then nature becomes operative. And if the polymorphic nature is harmful, then the polymorphic gene is eliminated by the nature. And if the polymorphic gene is useful, then it is selected by the nature. And in this way, the genetic polymorphism is provided a tool for the evolutionary effect. Now, as I think that you all understand properly what is the role of the genetic polymorphism in the event of evolution. And now I am giving you 1 or 2 minutes. If any viewer or a student has any question, then you give me message. If you have no any question, then give me okay. Now I am going to terminate my live stream.